Hello everyone, welcome to the course on medical biomaterials. We are going to talk about sterilization and device failure. Sterilizing a material is very important, whether it is for a short duration like your catheter or medium duration like cardiovascular stents or even total knee replacement joints and so on actually. Okay. So, if devices do not get sterilized properly, infection is being carried inside, which may lead to biofilm formation and finally rejection of the material, and the material has to be explanted out. So, sterilization is extremely important. Then we are talking about a biomaterial which is made up of metal or a polymer or a ceramic. So, we cannot follow the same sterilization procedure because a polymer might not be able to withstand very high temperatures, whereas metal will be able to withstand high temperatures. So, we need to have different strategies. So, we are going to look at some of those in this particular class. So, sterilization of implants. So, materials implanted in the body or human or an animal must be sterile to prevent infection, which can lead to serious illness or death to the host, number 1. Number 2, if we are using expensive instruments, we do not want to throw them out. So, we would like to use them, recycle them. So, we need to sterilize. So, whereas if it is very cheap instruments like your catheters or even your syringes or needles, it can be disposed, but very expensive instruments of course you need to sterilize. Okay, these two are good references to have a look at if you are looking at materials in medicine and issues related to sterilization. Have a look at these two. Okay, so we need to have a validated process because every time we cannot use an ad hoc sterilization procedure. So, it has to be repeated, it should be rendered a product free of all forms of viable microorganism. Okay. So, sterilization has to be a validated process. Disinfection on the other hand is destruction of pathogenic and other kinds of microorganisms by thermal or chemical means. Okay. So, it destroys most recognized pathogenic microorganism, but not necessarily all microbial forms like spores, because spores may be able to last longer and they may be able to withstand high temperature. So, it might kill many, but not completely. Okay. So, it depends upon the threshold, the lower limit. So, if you look at categories of medical devices, one is called the critical, semi-critical, non-critical. What is this critical? Like sterile tissue or vascular system, surgical instruments, cardiac, urinary catheters, implants, they are supposed to be critical systems, whereas semi-critical materials which are in contact with the mucous membranes or non-intact skin like endoscopes, respiratory therapy, anesthetic equipments, diaphragm rings, and then non-critical will be mostly bedpans, blood pressure cuffs, crutches. So, they just come temporarily in contact and generally it will not be in contact with the body fluids much. So, they can be called non-critical. So, the critical is that which is going to be in contact with the body fluids, may be kept inside the human system. Okay, that is the critical one. So, there is something called sterility assurance level. This is the generally accepted minimum sterility for implants. Generally it is called a probability of 10 power minus 6, that means 1 in a million that the implant will remain non-sterile, 1 in a million. Okay. So, there is still possibility of 1 in a million. So, if a hospital conducts 1 million surgeries, if you are following this SAL, sterility assurance level, then the chances are there could be 1 in a million which will not have a sterile biomaterial. So, how do you determine this sterility assurance? So, we determine the number of viable microorganisms on an implant before sterilization. Okay. That means you take 10 or 30 samples, then we shake it, sonicate, wash off the microorganisms from the implant into the fluid, then determine the colony count, it is called number of colony, by growing them in agar, incubating it over 24 hours. These are very standard procedures. Then after that you follow your sterilization procedure and again repeat the whole thing and see what is the effect of sterilization on the microbial kill rate. Suppose I am looking at time, should I do for 10 minutes or 20 minutes or 30 minutes or 40 minutes sterilization. So, I can do at different times and see what is the percentage of organisms that got killed. So, I plot a graph and then from there I decide what should be the time necessary for me to do the sterilization process. Okay. So, that is optimizing with respect to time. Same thing we can do with temperature also. Suppose I want to think about should I use 80 degrees, 90 degrees, 100 degrees. So, I can do the sterilization at various temperatures and then see what is the killing rate and then decide on what should be my optimum temperature. Okay. The easiest is steam sterilization. This is the oldest method, safest method, it is very cheap. That means we do it for about 15 to 30 minutes, 15 psi. So, we can reach about 120 degrees. It kills microorganisms by destroying metabolic and structural components essential for the replication. So, advantages, they are very efficient, fast, simple, no, it does not leave any toxic residues. Disadvantages, of course, we cannot use it for packaging material, we cannot use it for polymers, adhesives, materials which may melt or soften. That means if Tg is less than 121, then the material will get deformed if I raise the temperature to 121 C. If it is a hydrophilic material, it may absorb moisture. If it is a biodegradable or decomposing material like polyesters, then steam sterilization is not a good method, but it is the cheapest and it has been. Of course, we cannot do very prolonged heating because the moisture of the steam sterilization process may damage the material. So, we may have to go to chemical sterilization, radiation sterilization, which can be done on heat sensitive or moisture sensitive materials. Okay, they are slightly more expensive. Radiation, what do? We use cobalt 60 gamma rays. So, it produces ionizing radiation of cobalt 60 isotope source. Okay. Then the dose, sufficient dose is focused on the material, then the material becomes sterile. Okay. That is called the radiation approach. Another approach is ethylene oxide. This also has been there for a long time. Ethylene oxide is a gas. Okay. It disrupts the DNA and it can be used for all materials. Of course, it takes long time, precondition, temperature, humidity, aeration is a problem. But then if you are going to aerate it, then polymers, if they are present, they may start absorbing more air. Okay. That is a big problem actually. So, ethylene oxide generally 50 to 60 degrees centigrade, it kills microorganisms including spores by alkylating proteins and DNA. So, they should have a direct contact with the microorganism, okay, because ethylene oxide will directly get in contact on with the microorganism and then do the job, unlike the normal autoclaving approach. So, this is very widely used, as it says half the medical devices are sterilized using ethylene oxide. Heat or moisture sensitive material can be done with this, compatible with a wide range of implant material, packaging materials, suture, intraocular lenses, ligaments, tendons, heart valves, vascular grafts. So, it is very effective, high penetration because it is a gas, it penetrates through the interstices and kills, compatible. Disadvantage, it takes much longer time, 16 to 24 hours, whereas steam sterilization we are talking in terms of 30 minutes. It is extremely reactive and flammable. So, we need to handle with care, can leave toxic residues. So, we need to aerate it completely to remove them. So, it has a lot of physical and health hazards. Contact with the eyes, inhalation should be avoided by the person who is doing this type of sterilization. If you look at the gamma radiation, cobalt widely used for medical products against sutures, drapes, metallic bone implants, knee, hip prostheses, syringes, wide range of materials are compatible with the radiation. Polymers like polyesters, polyethylene, polystyrene, polysulfones, polycarbonate Of course, fluoropolymers like PTFE is not compatible. It is simple, rapid, effective, can be controlled by proper dosing. Large and small materials can be done, cost effective, not toxic, but it is very expensive because we need to have a cobalt 60 source and we need to get a lot of approval from the atomic energy for handling cobalt 60. And there will be continual decay of the isotope, so we need to do a very long processing time so that all the cobalt 60 is completely decayed out. So, other high temperature techniques, dry heat, we can go up to 160, it is not a steam sterilization, that means we do not use steam, it is just dry heat, 2 hours, limited applications, oils, petroleum products, bulk powders, that steam and ethylene oxide can penetrate, because if I am having these steam, that is water can penetrate into this, or even ethylene oxide can penetrate into this. So, that is called the dry sterilization. Temperatures are high and it is a limited application. Then of course, we have low temperature methods like hydrogen peroxide plasma around 45 to 50. Of course, it is expensive because we are using plasma. So, this is a method good between autoclave and high temperature sterilization. Okay. Because ethylene oxide may leave toxic residual, so high temperature is better. Quickly sterilize most medical instruments material without leaving any toxic material, because hydrogen peroxide is a gas. It is activated to create a reactive plasma or vapor. The time is also less, 45 to 70 minutes. Hydrogen peroxide, of course, it is a very good antioxidant and it is a very good antimicrobial agent, so it can kill many spores also. So, you can use this for heat sensitive material equipment such as endoscopic equipment, that is called hydrogen peroxide plasma method. Then we have sterile processing systems where we are using 50 to 56 degrees 30 minutes. We use peracetic acid, okay, where peracetic acid, as you know, is an oxidant and a disinfectant. So, we can use this. So, it can kill microorganisms at low temperatures. Okay. So, we do not have to go to 100 in autoclave or 150 or something. So, it reacts with most cellular components to destroy. So, it is a sterile processing system using peracetic acid. So, what we do is we have to immerse the material or the instrument, okay, and if the material cannot be immersed, then we cannot use this method, and we do not need to have a packaging for the material also. Okay. So, it has got some disadvantages. Then there is another method where you are using high level disinfectant HLD. This is an alternative to glutaraldehyde, because glutaraldehyde also can be used as a sterilizing agent. Okay. In 12 minutes at room temperature, and it is very good even for mycobacteria including glutaraldehyde resistance colianae. Okay. So, here you are using high level disinfectant, broad materials compatibility of glutaraldehyde, requires no activation, minimum odor. So, surgical cameras, cleaning and drying before immersion, rinsing with sterile water prior to use, and so on actually. This is called a Sidax OPA solution. Okay, this contains high level of disinfectant. Okay, you can use glutaraldehyde also. Of course, glutaraldehyde after sterilization will give smell, so you need to have it left out so that all the glutaraldehyde vapor evaporates. There are some banned products, there are some brand products there, brand names for glutaraldehyde, Sidax Plus and Sidax. Okay. So, Sidax Plus contains 3.4% alkaline glutaraldehyde. So, 20 minutes 25 degrees, so we can take in lot of bacteria, even tuberculocidal and high level disinfection. The other one is Sidax activated dialdehyde solution. Okay. This contains 2.4% alkaline glutaraldehyde solution. So, you need to put it for 45 minutes 25 degrees centigrade. Okay. So, we can use it at a very room temperature, that is 20-25 degrees centigrade, and it has got a very high level disinfectant. Okay, so these are solution based. We have high concentration of sterilizing instrument disinfecting liquid, or we can use glutaraldehyde, alkaline glutaraldehyde of two different compositions. These are liquid based methods, so we can dip the material in that. Then hydrogen peroxide in vaporized form for sterilization, and then plasma is called a VHP MD series. Okay. Cycle time is 2 hours, operating temperature 30 to 40 degrees centigrade. So we use hydrogen peroxide, and as you know, hydrogen peroxide is a very good antioxidant, so it can kill. And then we use plasma, okay, radio frequency energy. It is very expensive. It of course takes care between steam sterilization and ethylene oxide gas sterilization. Chlorine dioxide, this is a liquid, so this is a liquid based sterilization, 25 to 30 degrees 6 hours. So how do you produce this liquid? Dilute chlorine gas with sodium chloride to form chlorine dioxide gas, and this gas is then exposed to the equipment in a sterilizing chamber. Okay. So we have sodium chloride, we have chlorine gas, so they produce this chlorine dioxide, and your equipment is exposed to this. Of course, material may get corroded because this chlorine dioxide is extremely corrosive. We have oxygen and chlorine, both can corrode metals. Okay. Pre-humidification of chlorine dioxide is also required. So that means we need to add little bit of moisture into them. So lot of different methods. Of course, ozone. Ozone, as you know, is a very good oxidant, so it can also sterilize. It destroys organic and inorganic matters. Generally 60 minutes. It is also very corrosive. It may damage moisture sensitive equipment. It penetrates membrane cells, causing them to explode. Okay. Of course, other radiation sterilization, X-ray, E-beam, that is electron beam radiation, high dose of electrons in a narrow beam. It has got very limited penetrating power, less than gamma radiation. Of course, the radiation can change the properties of the material, especially plastic. They may lose some of their mechanical properties, okay, even optical properties. X-rays, again same penetrating properties as cobalt 60, faster, more flexible, more environmentally friendly, is good for penetration. It also helps in thoroughly treating the surface and interior of a product. Okay. So large number of methods we saw. We have the steam, we have the dry heat, then we have the ethylene oxide, which is widely used, and then we have different types of liquids. Okay. Glutaraldehyde, chlorine dioxide, okay, hydrogen peroxide, then disinfectant, high concentration of disinfectant, where the material is dipped in, and so on. But of course, the ethylene oxide takes care of 50% of the biomaterials that needs to be okay, sterilized. The steam sterilization is cheap method that takes care of many of these products. Of course, that is not very suitable if the material is heat sensitive or it starts absorbing moisture, then we may have to go for glutaraldehyde, other type of methods. Of course, the smell of glutaraldehyde has to go, so it has to be left open for a long time, and so on. So different methods are being practiced in the area of biomaterial, because material before implanted into human or animals needs to be completely sterilized. It should be free from bacterial infection. Okay, so device failures, devices fail. Many reasons are there for the failure of devices. Okay, we saw some of them during the course of our lectures, and they could lead to either debilitating effect or very serious even death. There have been many examples of device related deaths as well as permanent damage to the human. Poor sterilization, of course we saw that if the sterilization is poor and you have a bacteria entering this the human system, it could lead into biofilm formation, infection, inflammation, finally rejection of the material. Failure of electronic controlled instruments. Nowadays a lot of electronics is being used, your pacemakers, you have left assisted ventricular devices, okay, artificial heart, so many devices, and nowadays defibrillator, they all have some electronics into it, there will be some controls, okay. Failure of these electronics, that could be one problem. Mechanical failures, like we use catheters, we use sutures, they just break and the internal fluid may pop out, or infection bacteria may go inside. If you are using a urinary catheter and it breaks because of mechanical, you are going to have a lot of body fluids getting mixed up, okay. That is the mechanical failure. And of course diaphragm valves can have mechanical failure, and so on. Actually your vascular grafts can have mechanical failure, your dental implants can have mechanical failure, your orthodontic materials can have mechanical failure. Mechanical failure could include the stress related, or it could be compression related, or and so on actually. Environmental stress cracking, bimetallic corrosion, stress corrosion. Metals have one big problem, the corrosion, different types of corrosion, bimetallic corrosion, stress corrosion, heterogeneous distribution of elements corrosion, and so on actually, because the body fluids contain lot of inorganic salts, pH conditions are very acidic, so they are all very easy to get corroded. So the corrosion is a big problem, especially in orthopedic area as well as in dental area. So wear, fatigue, like I said, diaphragm valves, joints, knee joints, they may undergo fatigue, wear, and the ball and socket, overload. So you are putting too much load, so the materials could break or crumble or crack because of overload. Stress shielding. So we have stainless steel or titanium implants going parallel to the bone, so most of the load is taken by these metals, so there is a lot of the bone loses its the load carrying capacity, so stress shielding can happen. Biodegradation. So if you have polymers, they may start degrading because we have enzymes like lipase, esterase inside the body, which may slowly degrade. Of course we have oxygen also, so there could be oxidation taking place, so polymeric material may start degrading, so that is a issue. Once it starts slowly degrading, you may lose its mechanical strength, it may lose other properties. Absence of osteointegration. So if you have material which do not integrate, so they will remain separately from rest of the surrounding, of course it may they may loosen out. This can happen in teeth, this can happen in the orthopedic area. So loosening of joints because they do not integrate with the surrounding tissues. Infection, biofilm formation. This is a very serious problem because of poor sterilization or because of practices. Bacteria may enter which may get multiplied, it may get attached on the surface of the polymers and lead to biofilm formation, infection, rejection. We studied a lot, and the early rejection of biomaterial, the first few days, a week, is predominantly because of this biofilm and infection. Okay, that is why generally large amount of antibiotics are given to the patient to prevent bacterial infection and biofilm formation. Okay, monomer additive leakage. Okay, this is also very common. Dental acrylic acid leakage is a problem. Rest of the body you can have the polymeric plasticizers and other material which may be leaking out. So, that could be toxic to the person actually, like additives. Of course, even metal on metal interaction or wear could lead to release of metal debris, okay, which could be toxic. Sometimes patients have nickel toxicity or cobalt toxicity and so on actually. So, that is the metal ion leakage, especially that is a problem in artificial knee joints. Okay, so a lot of issues are there because of which biomaterial can fail. So, one needs to consider all these aspects, okay, when you are designing a biomaterial, be it metal, be it a polymer, be it a blend, or be it a ceramic. And of course, dangerous defective medical devices, faulty surgical instruments, implants, pacemakers, processes and so on. You can have a look at this website. It shows you some real examples of a list of recalls of medical devices by various companies because of the faulty designs. It is very interesting to read some of these problems. Okay. So, FDA also had observed many device failures, recent medical device failures, and they have listed them like inadequate sterilization. As you can see, sterilization, bacterial infection is the most important. Inadequate sterilization for an orthopedic surgery tool, rust in an injection, ventilators with defective components, infant resuscitators with assembly error. So, if you have multiple parts which needs to be assembled, there could be assembly error, guide wire with flaking coating. So, we have metal guide wires, we may coat it with oxides to prevent corrosion, okay. Then those could be flaking out. Tracheal tube that kinks, tracheal tube should not kink, because if it starts kinking, the area for the liquid to flow decreases, so the liquid might not flow at all. Unclear labeling, okay, that is another problem. If the label is not clear, which can lead to a surgeon making a mistake, okay, either putting it in the wrong direction or the wrong product, okay. So, these are some device failures that FDA had observed, okay, in their recent, okay, and this you can see it from this particular website. Again, it is very interesting to have a look at them. Okay. So, let us conclude on this course, the 20 hours course on medical biomaterials. We have had about 40 lectures, we have covered a lot in the past 40 lectures. So, the biomaterial is any substance, synthetic or natural, or combination of this, okay, used for some time or for a very long time inside the human body. It could be a part of the human body, it treats, augments, diagnoses or replaces. Diagnosis means it could be a biosensor or something, or replace any of the tissue, okay, or organ or function of the body. That is called a biomaterial. That is how it has been defined. Of course, a drug is not a biomaterial, whereas if you have a drug delivery system, the polymeric system which is carrying the drug, that is a biomaterial. Okay. So, we have been spending a lot on this particular topic in the past 40 lectures. Okay. So, we looked at metals, polymers, synthetic polymers, different types of synthetic polymers, natural polymers, polysaccharides, proteins. Then we looked at blends, that is polymer combination. Then we looked at inorganic materials, ceramics, calcium phosphate, calcium sulphate, okay, alumina, then the silicates, glass, that is silica glass. Then we looked at composites, combination of carbon reinforced polymers, okay, metal and glass impregnated polymers and so on. Okay. So, we looked at large number of these. Then we looked at the morphologies, different types of morphologies. We also had some demonstrations of experiments, how to prepare beads, how to prepare nanoparticles, how to prepare films, how to prepare electrospin fabrics, okay, how to prepare polymer gels. So, different morphologies we saw experimentally. Then surface modification using a dip coating method, using a spinning method, how to modify a polymer surface or a metal surface. So, we can have antibiotic or antibacterial material coated on surfaces. So, again we looked at it experimentally as a demonstration. Okay. So, we studied the material and blood interaction, material cell interaction, system response like inflammation, okay, a large number of biological functions that take place when a material is implanted into the body. So, we looked at all those as well. Okay. So, this course you can see it is very interdisciplinary. We had problems related to mechanical engineering, problems related to biology, and some problems related to polymer chemistry. So, here we worked out a lot of problems. We had lab demonstration. We looked at fundamental concepts in mechanical engineering and polymer in biology. We looked at what is this biofilm, inflammation, blood coagulation, etc. Then we also studied polymer chemistry. I spent a few lectures on analytical tools, because biomaterials has lot of analytical tools used for looking at the surfaces, looking at the bulk, looking at changes in their chemical properties, and so on actually. So, a lot of analytical tools are used. So, we looked at them, and then we spent short time on animal trials, how these animal trials are conducted, what are the issues in animal trials one needs to consider, what animals are used for what type of study, and so on. So, the whole course has been a combination of engineering, biological science, analytical chemistry, and polymer chemistry, and I hope you enjoyed this entire course. We also had a lot of assignments at the end of each week, which was supposed to help you to recap what you studied in that particular week. So, that should have been very beneficial for you, and you can always use these video as your future resources as well. So, whenever you have any doubts or you want to refresh certain areas, you can go back to that particular video and have a look at them. And of course, we also will be having a final exam where you can test your knowledge and your capability based on what you have learned in the past 40 lectures. So, I hope you enjoyed the course and you benefited from the course. Okay? Thank you very much.